Some of you will have been exposed to it and others not. I got my introduction through a summer course that's been taught here for several years. I have my phone on today because that call just could come, so sorry for the interference. This summer course that the MVZ co-sponsors and the UCMP is an amazing opportunity with two of the world's experts, Miriam Zeldich and Don Swiderski. And they like coming, and if the funding's there, they'll come back. So every summer has been this opportunity for years to take a crash course for free in geometric morphometrics, and I recommend it. It's a very satisfying pursuit, this. It's aesthetically satisfying. It's the old Darcy Thompson tradition, you know, brought into the digital age. And incredibly powerful analyses. With large sample sizes, you can get very, very robust support in many cases. And the software that I used was from this IMP series that was developed by those authors that I mentioned who teach the summer course, as well as a new program that is outstanding as far as I can tell. Morphogy, that's Chris Klingenberg who authored that, and then just statistical analysis and jump. In addition to those geometric morphometric shape analyses, I have information on the relative areas of the post-canine teeth for about the same number of individuals, as well as the traditional metrics, you know, one-dimensional linear caliper measurements, however you want to call them, taken on both cranium and mandible. And these are used for standardizing some of the other data, and I use them in other contexts. These chords that I chose to measure generally are ones that have been shown to be functionally relevant or are known as good basic data with which to correct other data in terms of size. So jumping right into the 2D geometric analysis of the group as a whole, this is a canonical variance analysis broken down by family for the carnivora. It kind of looks like a paintball shotgun, but in fact there's a lot of structure in there with 11 distinct canonical variants in an assignment test. You take any one of these mandibles and it's assigned to the correct family with less than 5% misclassified, more like 2.5% misclassification rate on those assignments. So that the phylogenetic information is coming out at least at that level of the family. And in mammals, family is a great group, you know, it's still a great grouping. Forget it in terms of a ranking context if you want, but those are still good clades, and it's a very appropriate level of analysis for a lot of these things. And it's that level of analysis and above that I'm keeping this approach here. I won't be talking about too many interspecific comparisons. But the mandible, which you can see in the landmarks here, I have, as one of those mandibles, gives you holds that phylogenetic information strongly, which is all well and good. But if you're interested in diet, you can see maybe how a dietary signal might be overwhelmed by the phylogenetic signal in that data. Another issue is body size disparity and allometric effects. You have this incredible range of almost 7,000-fold difference between a little least weasel and a big polar bear, and the sea otter, itself, which is a mustelid. We have two mustelids here with a great range of body size in between these two. So you also need to contend with that because there may be shape effects that are a consequence strictly of size, allometry, and there's a pretty straightforward way of dealing with that, whereby you take your estimate of size. Centroid size is the estimator that's used for most geometric morphometric analyses, the dimensionless variable of size based on the distance of the landmarks from the centroid. So when we use that as our estimator of size and regress shape on that, these coordinates when superimposed through Procrustes superimposition, I'm not going to go into the technicalities too much. When you regress that, you find that there is a significant relationship, and then you can you can take the residuals from that relationship and thereby and factor that out from the Procrustes coordinates and thereby handle, correct for size in your analysis of shape. So in the subsequent analyses that I'll run, I've done that. I'm using the residuals off of this relationship, so they're size-corrected data. And a plot like this represents the deformation, the movement of these landmarks, in this case relative to centroid size. So as you get bigger, this is the kind of deformation that takes place with this landmark, which is just here, moving downward relative to this landmark, which moves posteriorly, where these two are moving further apart. So what you're getting with increasing size is an increasing depth of the corpus of the mandible, a posterior deflection of the coronoid process, and other changes. So that's how you start to read a diagram like that. And I'll be looking at a couple more of those. So my interest is primarily in the form-function relationship here of form and ecology and diet. And so a priori, I assigned these things to dietary categories. And that's always funny business, assigning creatures like this to distinct categories. None of these things is a radical specialist in any really strict sense. These things are all opportunistic to a degree, but they do fall out into more or less good categories, most of them. And so I'll show you my categories quickly, and they're in order of increasing hardness of diet, which is admittedly subjective way of organizing this information. Ideally, I would have information on the actual hardness or softness of the diets of these creatures, measured however one might in the field. That data is not available for, at this scale yet, but someday it should be, and so you should be able to use that, good quantitative information later. For now, I'm just using a ranking system, with their softest object feeders being these earthworm, quote-unquote, specialists. Earthworms are a very widely used resource in the mammal world and for other tetrapods, of course. They've gone unnoticed a lot because they don't show up in the scat typically. There are means of earthworms have little bristles on them that if you're really careful, you can find them in the scat and recover them and actually generate good information on how much was consumed. But it's largely overlooked, and they're an important resource for lots of badgers. And then a couple of specialists, this great little vivarid from Southeast Asia. There's a couple excellent specimens in the collection here that Jim brought back some day ago. Another Madagascan carnivoran that's gone to eating earthworms. And they develop this long, attenuated mandible, very gracile, with a posteriorly deflected coronoid process. Just these long, break it on the table kind of dentary. Myrmecophagy, ants and termites, social insect eating. A few carnivorans are quite dedicated for that. The aardwolf, which is a hyena that's become a dedicated myrmecophage, almost lost its dentition completely. The bat-eared fox, great little canid from southern Africa, and the mongoose, for example. A couple of frugivores that just get bizarre. This really sort of goes toward a primate model. The kinkajou with its prehensile tail. This guy has a prehensile tail too. The binturong from Asia. Both the only really dedicated frugivores in the carnivorans. Although a lot will eat fruit, these guys are pretty much ripe fruit specialists. Take that with a grain of salt always, please. Omnivory, I distinguish between two types of omnivory, hard object omnivory and soft object omnivory. They're really different. Where these guys, these soft object omnivores, tend to rely on softer fruits, insects, snakes, amphibians, things like that. And they really, there just aren't hard components to the diet, whereas these hard object omnivores are your true omnivores of your raccoon, your common raccoon, and most of your bears, ourselves, pigs, baboons, things like that. A couple of piscivores. Great convergence here between Asia and Africa, and two piscivorous vivarids. Get these little sharp teeth in this Asian Senegale. and a pretty gracile mandible. Carnivores, so we speak of carnivorans and carnivores. I think you've probably gathered to distinguish the ecological term from the phylogenetic term. The categories continue. Hard-shelled invertebrates and small vertebrates, these creatures that combine those resources into a diet. Bamboo, I'm afraid I'm trying to coin a new word here because there's no word for it. And I'm sick of saying bamboo eating. Graminivory doesn't really help because that has different associations, even though bamboo is a grass. And that's about as good as you get. There are no Latin or Greek roots for the word, apparently. I guess they didn't know of bamboo in that period or didn't have a name for it. I don't know. It seems unlikely. But anyway, if someone's got a better idea, we need something for bamboo. I eat bamboo. And this is a phenomenon of the giant and red pandas. Hard-shelled invertebrates only. This is a separate category from this. These guys do a lot of different things, including a lot of fish and rodents and things. But this is our sea otter. Our very own sea otter is a really dedicated durophage. It eats almost no fish. There are populations that will, at particular seasons, rely on some of the slow-moving demersal fish. But they are pretty dedicated. They're almost exclusively dedicated to urchins, crabs, bivalves, abalone, things like that. And then your osteophages, your bone crackers. And I include the wolverine in that. The wolverine has been called the hyena of the north. It's a great scavenger, caches food for the winters, and thus relies on frozen carcasses for much of that season, and does a lot of bone cracking. It's a good osteophage. You might want to quibble and say, well, it should be in carnivore in the carnivore category or something, but this is where I started with that animal, and it looks like it's working out that way. Yeah, so this is the sense in which we can compare the shapes of these different groups on the phylogeny. So these shape deformations are this target taxon in relation to the root of the carnivore tree, the carnivoran tree. So you've reconstructed the basal nodes in the carnivoran tree through the square change parsimony process, I guess, is what Morpho-J is using, to reconstruct those ancestral nodes. And then you look at the deformation of the target tip taxon in relation to that ancestor. And when you look at something like Crocuta, the spotted hyena, the species that lives in the hills of Berkeley, and compare that to Gulo, the wolverine, which now we know lives in the mountains of California as well, based on some photographs that came out this summer, confirmed that it's here. You find, and this being a caniform and this being a feliform, totally separate on the tree, you find a very strong similarity in shape. If you take any particular landmark, say landmark 5, the end of the tooth row, you see relatively similar processes of deformation. If you could see that, I don't know how these grids are coming through. So one quibble I would have with Morpho-J is these grids are very faint. But the program allows these beautiful comparisons of ancestor to tip. And here it is for Aonyx capensis, the Cape clawless otter, one of these HSI and small vertebrate consumers, and a mongoose, again, a caniform and a feliform. And you can see some of those similarities in deformation between the two taxa. Regressing shape on dietary hardness according to those categories, you basically have a situation where increasing dietary hardness according to those categories is correlated with a change in shape in some sense. This is the sense where an increasing that increasing axis leads to these deformations, and these are the silhouettes that represent what's happening in that deformation. These are the descriptive ways you use these technologies to analyze shape. It's often you don't have to have, when you're shooting your landmarks, you don't have to have a priori expectations about what's going to be a useful parameter like you typically do when you're using your calipers. You expect this will be a good measurement and I should put it in. With geometric morphometrics, you just digitize, and then the important elements in the shape of the organism come through in the end, ideally. So it has that advantage as well. So call it fishing if you want for ideas, but it seems to be a perfectly valid way of generating ideas and hypotheses by shooting and watching the shape change. So I'm going to try to, wind this all together conceptually in the end, but we'll see. I'm going to be transitioning a little bit here. These are the hardest object groups, right? The osteophage, your hyena, the bamboozivore, your giant panda, and your HSI durophage. In the strict sense, the sea otter. Although they share similarities in the overall shape of the dentary, insofar as I've looked at it, they have these serious differences in the dentition, where the sea otter has these puffy round teeth that look like they've been inflated with air, whereas your bamboozivores have a highly crenulated enamel surface that's used for repeatedly breaking down bamboo, the woody bamboos, whereas this is just causing a fracture, and then it's really not doing too much chewing. This guy's even doing less chewing. A hyena tends to break the bone into bits small enough with these catastrophic fractures and then just swallow. And they use their neck muscles a lot in this process as well, with these massively hypertrophied musculature for doing so. So in spite of the similarities in the shape of the dentary, you have very different solutions to these different diets. And that's something I'll be focusing on soon, is looking at the topography of those tooth rows to try to get a better distinguishing function for these hardest object feeders. So I want to, there's no substitute for these kinds of things, so I want to show you a little video of a couple of these animals before I get into dexterity things. Got some volume. Just to give you an example of what this creature is like. This is one of my focal animals.
Prefers catching crabs to fish, but it finds the slow-moving puffer irresistible. But the puffer's apparent vulnerability is misleading. Inflating itself like a balloon makes it much too large to swallow and raises thousands of tiny prickles.
I just wanted to give you an idea of that animal. Doesn't really shed much light on its diet there. It's really I think it gives a good sense for the animal. In terms of, there were a couple of shots there where it was manipulating the puffer on the surface of the water. It's got these unwebbed fingers with these broad apical tufts on the distal ends, with which it's primarily searching in the muds and banks and under stones for crabs, which are a very common resource in its range, in the fresh waters in its range. I mean, you have puffer fish and crabs in these fresh waters, not in North America anymore. And Aeonyx has this huge skull, a tremendously robust skull, and the short ears, of course, the long whiskers for murky waters where it typically dwells. It occupies as a different species, the Congo Basin and heavy swamp forests, very well known to show the same patterns. So you have this commonality in brain structure, and a study of brain size in South Africa showed that in overall size, the Marsh Mongoose and the Qualis otter have the biggest brains. And they linked it to their diets. Some of these things that I mentioned that I've been studying, the crabs and mollusks. They linked it to an easy-to-get, highly nutritious diet. I'm exploring more the connection to the hands. And I think that has probably a stronger relationship to overall brain size, that hypertrophication in relation to sensory motor dexterity. These different threads just haven't been tied together. They're just sort of hanging around in the literature. So that's my focus with the dexterity studies. Here, that connection between just the massiveness, the robusticity of the skull and brain size is what I'm testing here. And you have to jump through a bunch of hoops to do an analysis like this, because I'm interested in corpus robusticity, which is the cross-sectional area of the mandibular corpus, which is a estimator of bending strength, as well as just the overall area of the post-canine dentition in relation to the size of the brain. This is a sea otter that has pigment from its diet that has gotten into its cranium, an Alaskan otter. So, brain volume, occlusal area of the post-canines, and corpus robusticity first need to be standardized for size in relation to metrics that are appropriate, like the length of the cranium or the length of the dentary in the case of the corpus robusticity. So you generate these residuals from this relationship, and then you use those residuals in an independent contrasts analysis. So you're doing an independent contrasts on the residuals of those relationships. I think I've got all this right. These are the hoops one needs to go through. And then you can look at these corrected for size and phylogeny values of brain size in relation to corpus robusticity and tooth size. And there does seem to be a relationship between just having more massive teeth and a more robust jaw and having a big brain. And that's something I'm going to be trying to unpack further. So the whole background, I started working on African otters before becoming interested in paleoanthropology, but when I first started learning about Australopithecines, the morphology of these robust Australopithecines struck me as similar, akin to the shape that you see in, I don't know who I started with, but I started with Australopithecines. Anyway, the shape seemed to me akin in the Australopithecines to that of the durophagus carnivorans. That's a connection that is kind of was kind of nuts, and so I sat on it for a while before starting to talk about it, so I had my ducks in a line, because these ideas on the robust Australopithecines, these hominids that coexisted with early Homo, our ancestors, for 1 million years in Africa, these guys have been studied for a long time. They're well known. They're one of the best studied fossil mammals in the world. Going back to the 50s with John Robinson, all these ideas on what kind of diet, what kind of ecology these robust Australopithecines might have had: grass seed, bone cracking, bamboosivory, frugivory, or cracking hard nuts, just an omnivore like a bear or a pig, underground storage organs, tubers and roots and things like that. So all I've done is add to the list, and calling it the durophage ecotone model, to suggest that, to hypothesize that the robust Australopithecines were actually, in part, foraging for hard-shelled invertebrates and ecotones, in addition to a general, primate foraging strategy of get what you can, including a lot of fruit. So it's in testing that. This is just an example of one of these robust Australopithecines from East Africa. This is from Olduvai Gorge, the one that put the leakies on the map, with its incredibly massive post-canine dentition. This the smooth, flat construction to the teeth, with the hyper-thick enamel of these teeth. It looks very much to me like what you see in something like a crab-eating raccoon, with a curvature, concavity of curvature that wears flat, with and all the metrics line up, with the overall corpus robusticity and the structure of the musculature. So that's the morphological background to the idea, and my means of testing have primarily been biogeochemical, with trace element and isotope analyses. So the suggestion is that the robust Australopithecines, the raccoons, the otters, all these things are linked in some fashion by an emphasis on dexterous foraging in land-water ecotones for hard-shelled invertebrates that has correlative implications for the structure of the brain and a whole suite of natural history phenomena. So that's what I'm trying to do. thanks to museums and their curators, thanks to faculty here in IB, and thanks to undergrads who have been contributing a lot to this work, both here in the museum, up in the lab, and in the field, and the museums, and for financial support. Thanks.
Thank you.
Okay, Alan's left us a couple of minutes for questions. We'd like to leave it. Phil.
You've convinced us about all these behavioral adaptations that are alternatives to dealing with hard objects, in addition to the jaws. And somehow you've got to factor that into your jaw analysis, it would seem.
Yeah. How you would quantify it, I'm not sure. But the fact is that in the case of the Marsh Mongoose, if I'm understanding you, though, and for example the sea otter, they're both tool users that are relying on a technology to deal with these hard objects. But they also have this massive construction in the skulls. So the one doesn't obviate the other. They both are adept at using tools for the hardest objects, and then have the massively constructed skull for dealing with
So
everything.
they're not in conflict.
They're not in
obviously.
conflict, but I don't know how you would quantify the technology if you wanted to put it into a formal analysis. Because we assume that the Australopithecines, these robust Australopithecines, were also good tool users. Although there's a lot of debate, we assume that they're at least as good as chimps at such
I wonder, how much developmental plasticity is there in some of these landmarks? So if you feed some of these, I mean, I'm thinking of raccoons getting in the back door of my house, eating my dog's food, that sort of thing.
Yes, probably a lot, and this would be a great way to investigate. I should have mentioned I focused only on adult skulls with all sutures fused and all teeth erupted. But this is the technology really for capturing those kinds of allometric changes with age, but as well as
With diet.
different diet and different populations. One suggestion has been made to me to look at raccoons from out at Point Reyes that are foraging in the shallows for hard-shelled invertebrates so commonly, whereas yours are eating your garbage. You know, if you could get two populations of such a in such a case, that would be a great comparison. My analysis tend to be at a higher level than that, so I haven't gotten down there, but it's all a great use for this.
You ignored a big group of carnivores, the marine mammals. Are they just noise in the system, or are there any insights to be gained
They're
there?
quite noisy. Yeah, I should have said that too. Non-pinnipeds here. Pinnipeds are nested within the caniformia, your seals, sea lions, and walrus. They're complicated. They're funny. You know, they have a homodont construction where you lose the differentiation between premolar and molars oftentimes. It's dietary information is very hard to come by on what they're doing down at however many fathoms. So like a lot of these analyses, it's focused on terrestrial carnivorans, and we pretend that pinnipeds don't exist. Yeah, I don't.
No pressure. No pressure. No pressure. And he's actually got an interesting history of putting together information from bones and ecology and morphology and evolution and paleoanthropology, and he's weaving it all together into a really cool project about paranthropus, which I don't know if you'll touch on today or not, Alan, but as an outgrowth of that, he's been spending a lot of time in Africa, looking at everything from otters to crabs, and I guess today he's going to tell us why with his talk on dexterity, durophagy, and brain size in carnivoran mammals. And we actually owe an extra special thanks to Alan today, because his wife is sitting at home right now just about ready to have a baby. So if the phone rings, you may have to leave.
I do have my phone on, but she's actually at work, so we're planning on Sunday being the day. Everyone's available on Sunday, and so we're sort of scheduling it then. Thanks, Tony. Yeah, so this tapestry that I'm weaving is sadly unfinished. And here, foolishly perhaps, I'm trying to reach beyond my thesis research into the data that I'm gathering for the next phase of work. I gave an MVZ lunch, I guess in fall last year, and focused on my biogeochemical research, the isotope and trace element research, with a focus on paleoanthropology, testing a hypothesis concerning one of our close hominid relatives from the Pleistocene. And this is all in the context of that research, but it's a development and an outgrowth from it. And I'll talk about some of that paleoanthropological information at the end of the talk. So jumping right in. Yeah, so I'm, telling stories here. Trying to start off the semester with some good natural history with the greatest clade on Earth. And which also makes for a very effective comparative context when asking questions about other mammalian or perhaps non-mammalian clades. The carnivora is divided into two great groups, the cat-like forms and the dog-like forms. And the cat-like forms in the Feliformia include the familiar cats, hyenas, mongooses. This is a Madagascan carnivoran, Eupleridae, the family that it's usually assigned to today. Mandinia, the basal member of this entire feliform radiation, a monotypic branch that lives in the rainforests of Africa. And the Viveridae, the genus civets, and the very numerous and speciose carnivorans of the Old World in that group. And then the dog-like forms, which are perhaps more familiar. Canids, dogs, wolves, and friends. Skunks, stink badgers, and friends. Red panda. The procyonids, which is a strictly New World radiation. Raccoons, coatimundis. The mustelids, a huge group, including the weasels and otters and badgers. Oh my. And the ursids, which, only 6 or 7 species of bear in the world, right? Much broader distribution previously used to be in Africa as well. So one of the reasons why clade carnivora is an exciting place to study at the moment is because we now have a pretty well-resolved molecular phylogeny, not for the group as a whole. This is stitched together from studies on the major families of the carnivora, but it's very close, and so the phylogeny that I'm working with in the background here is one that I've concatenated from these other studies. There's very good resolution with the feliforms represented here. Mandinia, as I mentioned, as a basal group, cats, viverids, genus civets, hyenas, mongooses. Then your caniforms with the basal lineage of the canids, the dogs, ursids, mephistids, and then the musteloids, procyonids, and mustelids. And I'll be returning to this a little bit when we get into using it as part of our analyses. So what I'm trying to stitch together here is, and the way I'm starting to talk about this is durophagy, which just means I eat hard things. Duro meaning hard and phagy to eat. HSI durophagy being perhaps a more specific kind of durophagy. With HSI, I'm just referring to hard-shelled invertebrates to distinguish it from other types of hard object feeding. So hard-shelled invertebrate durophagy as a biological syndrome or something like that. Something to capture the sense in which it's a total natural history phenomenon for the organism involved. At least is the way I'm seeing it, and what I'm trying to look at in terms of the analysis of the data. A big crawdad to represent your HSIs. These animals that consume these types of resources that are usually either in the shallows or in the ecotone between land and water, occupy particular types of habitat that are consistent. Their diets are HSIs. They seem to have similarities in their brains that have been borne out in various independent studies. The hand structure can be similar, and we'll look at that. with this loss of webbing between the digits. I mean, even your cats and dogs have webbing between their digits. Things like raccoons and some of these other creatures we'll look at have lost that and have converged in form, apparently, on this manus structure. And then in the skeleton, and I'm focusing on the skull here, but it extends to the postcranium as well. So this sense in which to be an HSI durophage entails these several things, apparently, is what I'm trying to get into. Shape analysis is what I'm really going to be talking about today, geometric morphometric analysis in 2 dimensions with a focus on the dentary. I'm just now starting to organize the information for the cranium, but here, this is just data on the dentary, which is the lower jaw, and it's a single bone. It's modular in its construction, but it's a single bone in the mammal, and in that sense, it's unlike the cranium, which is a much more complex structure. It's a neat structure to study, and it's very amenable to 2-dimensional analysis because it's relatively flat, and you can face the camera at the proper angle, and you can get a good 2-dimensional view of it, which is more difficult with some other structures. I've included almost 900 specimens in the analysis from 137 species of carnivorans. The ones that I had on that phylogeny are the ones that I've included. That phylogeny wasn't inclusive of all carnivora. I'm not sure how many species actually there are in carnivora, over 200 though. So I'm sampling over about 60% of the world's carnivoran species diversity. I digitized these 20 landmarks, but for the analyses I'm presenting here, I'm excluding some so that I can look at the carnivora in its entirety, because there are differences in tooth structure in the carnivora. For example, felids and hyenids don't have the second molar. It's permanently lost. And so you set up these landmarks in your digitization to be homologous across specimens. But in defining them, comparing a cat and a weasel, you have to choose landmarks that can be defined homologously between those 2 animals, in spite of the difference in dental formula. So, although I digitized 20, I drop out these 2 and define this as the end of the tooth row, and then I leave out these cusp landmarks because not all the specimens are perfect, and some have damaged teeth and missing teeth, and so what I end up focusing on is just the outline of the dentary, just that shape. And I'll look at it in this context of thin plate spline deformations. I won't go into the nitty-gritty on all that, in all that much detail on geometric morphometrics. I don't know, frankly, how informative they would be. What I do know is that the Madagascan carnivorans are very amenable to analysis. Some people also exclude those, but they present a great radiation that in many ways mimics the mainland Africa and mainland South America, just in the radiation on that island. So that's one that I've at least got in here.
Okay, well I think I'll need to draw to a close if people need to get to classes. So Alan, of course, is about, happily. Although finishing his thesis this year. Okay.
This semester.
Oh, this semester. Anyway, thank you again, Alan. That was terrific.
And I compared, I said it's one of my focal animals, and this being the other for the mammals. I'm comparing it with the Marsh Mongoose, which it shares almost its entire range with.
The marsh mongoose is a solitary creature. It feeds mainly on crabs and shellfish it finds in the shallows with its nimble and sensitive hands. This one is after mussels, which he will break by throwing against a rock. Failing to break it in the shallows, he carries it to a favorite anvil stone. Mongooses are sometimes caught and eaten by crocodile.
They do that quite well, and you can find on the banks of these water bodies one of these anvil stones or an old stump or even a glass bottle that has these sprays of shells around them. They'll just use anything they can. They rear up and hurl them down vertically. This is taken from stills of that video. Unlike some of the other mongooses, which have another hurling pattern that is fairly common, 6 or 7 species have been seen to do it, where they're throwing it back between their legs and they jump up a little bit and let the object go beneath them and behind them. Maybe Adillax has a more derived configuration relative to that, but it's its means of breaking open the hardest objects that it can't crack with its teeth, or that might damage its teeth. And again, this is another case where these resources are probably vastly underestimated in their diets, which have been so well studied based on scats, because mussels just don't come through the other end so obviously either. Otters, the big otter has been attributed, has been said to have made some of these anvil stations and the debris around the anvil stations, but probably it's all being done by Adillax. There's really no good evidence that Aonix does this. And then your familiar raccoon.
So you might say
in the New
that
World.
the raccoon sees with its hands. It's certainly able to feel the difference between a rock and a clam in a split second. If it touches a crayfish, which is armed with powerful pincers, then this second sight is clearly very valuable.
And then just goes to the teeth with
is not even affected by temperature.
the crayfish. Not that hard an object. Crabs and crayfish, if they're small, they're harder than beetles, but they're pliant. They're made of chitin, so they're capable of being crunched in the jaws. It's not like you need tools for those. It's just the bivalves that tend to lead to the tool use, and you see that in the sea otter, of course, too. So these three guys that I've just highlighted, your mongoose, your marsh mongoose, your raccoon, and your clawless otter are quite distantly related, but they share this structure in the manus, which is unique within the carnivorans. This was recognized a long time ago. This figure comes from 3 publications, Pocock's, a mammologist in the early 20th century, comparing the three. What I'm doing, and what I'd hope to bring into this talk, is a study of the osteology underlying this by dissecting these animals and as many other carnivorans to flesh out the context as possible, and looking at the individual elements underlying the soft tissues to see if there are any bony modifications associated with this obvious soft tissue modification and convergence. And alas, I didn't get it done in time. This is the crab-eating raccoon from South America. It's the focus for me of that handwork, and it's particularly robust in its skull and dentition. Here's an image of one, sister species to our northern raccoon, but just more extreme in the robusticity of its skull and the surface area of its dentition, and it looks to be somewhat different in the structure of its hands from what I've seen so far. I wanted to put a couple of herps in here. These are examples here in South America of a good durophagus herp, right? It's Tia Dracena. And maybe you're seeing sort of a consistency in the form of this skull with the massive bulbous dentition, the low slung deep mandible, the overall robust skull, and then an African monitor that also eats these guys are molluscivores primarily, and these guys are primarily eating crabs and some mollusks as adults. These are the skulls of Aonix and Adillax, the clawless otter and the marsh mongoose, again showing the robusticity of that you see in this type of feeding. And I've been looking at their fossil record, which goes back a few million years in Africa, and studying their biogeochemistry in relation to their prey. So a lot of my thesis work was field work involving collection of invertebrate prey items for these animals and sampling the tissues of these guys, primarily from museums and the occasional roadkill. I'm not out there collecting otters and mongooses, unfortunately. And trying to establish the biogeochemistry of that trophic relationship. But what I'm opening up now, and I haven't explored previously, is because in looking at these guys, it's always been obvious that they have big brains, and it's known in the literature in isolated cases that these same animals that are using their hands for foraging for HSIs also seem to have big brains. The old mustard seed technique was the one usually used, where you fill the cranium with mustard seed and then you pour it into a volumetric instrument and measure, cranial capacity. Rudinsky, who did so much important work on the carnivorans, including brain neuroscience of carnivorans, developed a technique using latex casts that he would then take a volume estimate from. Curators tend to be okay with those things, but mustard seeds in the collection aren't great. What Finarelli did was to adapt a simple ectocranial technique for estimating brain size based on studies of rats and primates that he found to be highly correlated with brain volume in specimens that for which that brain volume was known through other means. So I realized during the course of my study that this would be a cheap and easy way to get estimates of brain volume from the same specimens that I was already working on. So I went back to the museums and collected these 3 extra dimensions. And all you need is a height of the neurocranium from the base to the top of the cranium, not including the sagittal crest, the width, the widest points of the neurocranium, and the length. And this is the roughest because your landmark here is the frontonasal suture, but this isn't brain in here. The brain cavity really ends back here. So you are including boogies, not brain, right? So you are including non-neural tissue in there that doesn't seem to confound the analysis, since that length is so strongly correlated with the actual length of the brain case. I mentioned Rudinsky's studies. He found in independent studies of Adillacs and Aonics that they have hypertrophications in the region of the brain, the post-central gyrus, that's correlated with the sensory motor activities of the hands. And he also found that to be true in the sea otter here, our California sea otter, but found that it was arrived at through a different pathway. So you had 3 cases of independent evolution of a common structure in the post-central gyrus related to sensory motor dexterity. And the raccoons have been particularly well studied in this regard, and they are